The overall goal of this procedure is to generate 3D renderings of placenta villus tree fragments that are suitable for the analysis of their contained vascular networks.
This method can help us answer key questions in our field, such as why a child may be growth restricted, or what stress and how severe the stress might have been that led to preterm birth.
The main advantage of our procedure is that we are now in a position to immunolabel clear placental villus tree tissue and then visualize their vascular networks and use those visualizations to characterize and analyze these vascular networks.
The implications of this technique can extend into many diagnoses and therapies, since most life sciences rely on two-dimensional histological characterization.
Demonstrating the procedure will be Ms. Valerie Schwenk, who will be carrying out the immunolabeling and clearing of the placental tissue, and then Mr. Phillip Necaise will be doing the dissection of the placenta tissue and doing also the reverse clearing of the tissue.
To prepare for the dissection of the villus tree, first, rinse the formalin-fixed placental tissue with phosphate-buffered saline, and then place in a petri dish on the stage of a dissection microscope. To locate the villus tree, use scalpel and fine forceps to tear the placental tissue apart. Then, dissect pieces of the villus tree approximately 5 millimeters in length and transfer into a microtube containing 1 milliliter of phosphate-buffered saline. Replace the phosphate-buffered saline with a fresh one, and wait for 10 minutes with occasional swirling.